Historically, biopharmaceutical production has been a game of massive scale and glacial timelines. Companies built dedicated billion-dollar facilities to produce globular proteins, operating on a development cycle that typically stretched over 15 years from discovery to the pharmacy shelf. That rigidity creates a terrifying financial environment. In 2019, Biogen had to halt 2 phase 3 trials for an Alzheimer's drug after years of investment. Their stock price dropped more than 33% overnight, proving that under the old model, a single clinical failure can jeopardize the survival of even the largest firms. While those corporate risks were mounting, the underlying science reached a threshold. Decades of research into genomic editing and cellular engineering finally produced tools like CRISPR and CAR-T therapies, giving researchers the ability to reprogram a patient's own immune cells to hunt down specific cancers. This created a physical crisis. Living cells and sensitive genetic instructions cannot be mass-produced in the same giant steel vats used for standard chemicals. They are fragile, highly specific, and require an entirely different kind of hardware. The industry realized that inventing a miracle cure is effectively useless if the existing infrastructure is physically incapable of manufacturing it. The most prominent disruptor to the status quo is CAR-T therapy. Instead of a pill, the drug is the patient's own T cells, which are extracted, genetically modified in a lab, and then reinfused to attack tumor cells. Engineers build a chimeric antigen receptor, like this Yescarta model, with an antigen binding fragment to find cancer and signaling fragments to trigger a kill response. Comparing Yescarta to 2021's Abecma shows added co-stimulatory domains that keep T cells active for longer periods. But because these are living, aggressive cells, the safety risks are high. If the immune system overreacts, it can trigger cytokine release syndrome, a lethal level of systemic inflammation that requires constant clinical monitoring. To manage this, scientists are now building off switches into the cells. By adding an inducible safety gene, doctors can administer a specific molecule that triggers cell suicide if the treatment becomes dangerous to the patient. This precision extends to CRISPR-Cas9, which functions as genetic scissors, allowing for the surgical removal or repair of defective genes. This moves precision medicine beyond immune cells and into the treatment of inherited genetic disorders. When the drug product is the patient's own biology, the industry has to abandon assembly line uniformity in favor of microscopic, customized batches. The manufacturing response to this shift was a move toward miniaturization. Massive static factories are being replaced by continuous processing networks that are smaller, faster, and more flexible. In a continuous platform like the iSkid, harvesting and purification happen simultaneously without pausing for long in-process holds. This achieves a 20-fold reduction in vessel volume and slashes 18-month development timelines down to just 3 months. This smaller footprint has led to portable manufacturing. These are self-contained, automated production pods that can be shipped and deployed closer to hospitals and patient populations, bypassing the need for a central mega-factory. The logic of miniaturization has been pushed to its limit by the U.S. military. DARPA recently funded the development of a biomanufacturing platform roughly the size of a briefcase, designed to produce therapeutics on demand in the field. In this new landscape, production power is no longer measured by the square footage of a factory, but by the agility of the process. The COVID-19 pandemic served as the ultimate proof of concept for this accelerated manufacturing model. mRNA vaccines changed the fundamental approach to manufacturing. Instead of using a factory to grow viral proteins, the vaccine delivers genetic code that instructs the patient's own cells to produce the antigen and trigger an immune response. The speed of this platform was record-breaking. Moderna moved from the initial viral sequence to a physical vaccine prototype in 42 days, compressing a process that traditionally took 15 years into less than a single year. However, this speed creates a massive logistical burden. Because mRNA is inherently unstable, the raw vials must be stored in an extreme sub-zero cold chain to prevent the genetic instructions from degrading. To manage this, the supply chain went digital. Using the Internet of Things, distributors tracked multi-dose packages with integrated GPS and RFID sensors, monitoring the temperature in real time to prevent spoilage across the global network. The biopharmaceutical industry has completed a total transition. It has evolved from a slow-moving manufacturer of mass-market chemicals into a high-speed deployment network for on-demand biological code.